Assalamu alaikum, ladies and gentlemen. Good noon. I am Zunaira Said. I warmly welcome you all on the 7th International Conference on Applied Zoology 2024 on behalf of Organizing Committee ICAZ. I will be the moderator of this session, which include talks from the field of nutrition, toxicology, diabetes, and genetics. Let's we start our session with the talk of Dr. Sana Zahoor from Virtual University on the topic of process parameters for the optimization of endoglucanase production by Bacillus licheniformis using agro-industrial byproducts. Ma'am, please come and enlighten us with your valuable knowledge on the topic.
2024 for providing me an opportunity to present my work here. The topic of my presentation is process parameters for the production by Bacillus ligniformis using agro-industrial byproducts. Endoglucanase is an important enzyme that hydrolyzes cellulose by attacking on the amorphous region of the cellulose and give rise to more accessible free chain ends. Endoglucanase find application in many industries like food industry, where it is used in the clarification of juices. In textile industry, it is used in the biopolishing step and for the finishing of fabrics. In paper and pulp industry, it aids in the pulping process and improves the yield of the paper. In agriculture, it is used in the soil treatment, where it decomposes the organic material, thus improving the soil quality and nutrient availability. In biofuel production, it breaks down complex cellulosic materials into simple sugars, which are then used for the production of biofuels like ethanol. In detergent, it is used for the removal of cellulose-based strains, and in the pharmaceutical industry, it is used in the formulation of certain drugs. Different organisms have been reported to produce endoglucanase. These include bacteria, fungi, and plants. Different strains of Bacillus and Clostridium are potent producers of endoglucanase, while different fungi including Trichoderma, Aspergillus, Penicillium, Fusarium, and Neurospora are also good producers of endoglucanase. Different plants like Arabidopsis, Ziamase, Oryza sativa, and Triticum are also reported to produce endoglucanase. Among different microorganisms, bacteria are preferred for the production of endoglucanase because they have faster growth rates, they have simple nutritional requirements, they are capable of growing and producing enzyme of less complex media. It is easy to manipulate the genetic makeup of the bacteria, thus improving the yield of enzymes. They have the capability to produce high yields of active enzymes. Bacterial fermentations face less contamination as compared to fungal fermentations, and they have the ability to use a wide range of substrate for the production of enzymes. Solid yield of endoglucanase. As it uses agro-industrial residues, it is environmental friendly and cost effective. Solid state environment enhance nutrient availability. There are less chances of contamination in solid state fermentation as compared to submerged fermentation, and it uses less energy for mixing, aeration, etc. 16S rRNA gene sequencing is a preferred method for the identification of bacteria because it is present in all bacteria, thus making it a universal marker for the identification and differentiation of bacteria. It has conserved and hypervariable regions that allow the differentiation of closely related bacterial strains. Sequencing 16S rRNA gene provides high resolution data that aids in the identification of bacteria. It provides evolutionary relationships among different bacterial strains. It is a rapid and cost-effective method for the identification of bacteria. Endoglucanase producing bacteria were isolated from different soil, compost, and agricultural waste samples. Screening of endoglucanase producing bacteria was done using CMC agar plates and Congo red dye. The isolate that gave the highest production of endoglucanase was characterized on the basis of morphology, biochemical test, and molecular characterization was also done. In morphological characterization, colony characteristics and cell characteristics were studied. In biochemical characterization, catalyst test and oxidase tests were performed. In molecular characterization, 16S rRNA gene was amplified and phylogenetic analysis was done using MEGA11. For the optimization studies, effect of different substrates, diluents, pH, time period, temperature, and inoculum was studied on the production of endoglucanase. Total 24 different endoglucanase producing bacteria were isolated from 17 different sources, and bacterial isolate C6 isolated from compost sample gave the maximum production of endoglucanase, so it was selected for further studies. Regarding morphological characterization, the isolate formed cream-colored circular colonies with entire margins. Regarding biochemical characterization, the isolate was gram-positive and catalase positive, while it gave negative test for oxidase. 16S rRNA gene sequence of the isolate gave 99% similarity with Bacillus ligniformis, and the phylogenetic analysis showed the similar situation. Effect of 9 different solid substrate was studied on the production of endoglucanase, and rice straw gave the highest yield of endoglucanase. Effect of 6 different diluents was studied on the production of endoglucanase, and diluent D4 containing ammonium sulfate, potassium dihydrogen phosphate, magnesium sulfate, and calcium chloride gave the better production of endoglucanase. In addition, the effect of different diluent volume was also studied on the production of endoglucanase, and it was found that 10 ml diluent gave the maximum production of endoglucanase. Effect of different pH ranging from 5 to 8 was studied on the production of endoglucanase. The isolate gave maximum production of endoglucanase at pH 6.5, showing the slightly acidic nature of the enzyme. Regarding temperature, maximum endoglucanase production was obtained at 37 degrees centigrade. Effect of different time period was studied on the production of endoglucanase, and maximum endoglucanase production was obtained after 72 hours of incubation. Effect of inoculum was studied on the production of endoglucanase, and 10% vegetative inoculum gave the maximum production of endoglucanase after 72 hours of incubation. These are the references. Thank you for your attention.
Thank you so much, ma'am, for sharing your words. Now, I would like to welcome Dr. Fahmida Channa from University of Sindh to talk on the topic of prevalence of diabetic nephropathy in type 1 diabetic patients and its association with major risk factors. Ma'am, please come and share your knowledge with us.
Bismillahirrahmanirrahim. Assalamu alaikum. This is Dr. Fahmi Dachana. Today I am going to presenting my research on prevalence of diabetic nephropathy in type 1 diabetic patients and its association with major risk factors. Contents of presentation. Introduction. Diabetic nephropathy is a major microvascular complication of type 1 diabetic mellitus leading to progressive kidney damage and increased risk of chronic kidney disease and end-stage renal disease. Almost 1 in 5 people with diabetes will need treatment for diabetic nephropathy. Diabetic nephropathy is a leading cause of mortality in type 1 diabetes. Microalbuminuria, generally recognized as the earliest clinical feature of diabetic nephropathy, has a cumulative lifetime incidence of approximately 50% in type 1 diabetes and develop at a rate of around 2 to 3% annually. Diabetic nephropathy, heredity, glycemia, age, blood pressure, dyslipidemia, cigarette smoking, impaired renal function, diabetes duration. Let me talk about objectives. This study aims to determine the prevalence of diabetic nephropathy in patients with type 1 diabetes and to explore its association with key risk factors such as duration, diabetes, hypertension, and lifestyle factors, to investigate the relationship between the duration of type 1 diabetes and the occurrence of diabetic nephropathy. Materials and based cross-sectional study. Data collection was conducted from January to December 2018 during routine working days. Data were collected through self-developed questionnaire in English version. During visit, oral consent was taken from the type 1 diabetes patients and pre-designed questionnaire was filled out by asking questions to the medical OPD patients of both hospitals of Jamshedpur and Hyderabad. Questionnaire based on demographic, medical history, diabetes duration, blood pressure, and lifestyle factors will be collected through structural questionnaire and medical history or records. HbA1c levels and kidney functions, just like urine albumin to creatinine ratio, will be measured to assess glycemic control at the presence of nephropathy. Questionnaire. Now let me talk about the results. This figure shows the most of the patients were male, about 56 patients. 44 patients were female. This figure shows the age groups of diabetic nephropathy patients. More than half, it means the 60% of patients were age groups 26 to 30 years. 40% of patients were age group 20 to 25 years. This figure shows the family history of diabetic nephropathy. Around 60% patients were the negative family history of the type 1 diabetes, and the 40% of patients were the positive family history. This figure shows the glycemic status in a diabetic nephropathy patients. 72% of patients were poor glycemic control and 28% of patients were the good glycemic control. This figure shows the blood pressure monitoring in the diabetic nephropathy patients. 80% patients were hypertension and 20% of patients were non-hypertensive. This figure shows the smoking in diabetic nephropathy patients. Males, about 78% of males were smokers and 22% of males were non-smokers. This table shows the hypertension correlate with the stages of the diabetic nephropathy progression. 5% of patients were hyperfunction and hypertrophy. At the second stage, silent state, the 15% of patients were hypertension. Third stage, incipient stage, at the 30% of patients were hypertension. At the fourth stage, overtime diabetic nephropathy, 50% of the patients were hypertension. At the stage of the uremic or end-stage renal disease, 80% of patients were hypertension. Now let me talk about the conclusion of this results or research. The present finding indicate the prevalence of diabetic nephropathy approximately 30%. Over half of the affected individuals were male. The major risk factors of diabetic nephropathy in type 1 diabetes mellitus include gender, duration of diabetes history, poor glycemic control, and hypertension, all of which were found to have a significant association with the development of diabetic nephropathy. Recommendation. The finding highlight the need for the aimed at the controlling diabetic nephropathy. It is also recommended that the healthcare professionals provide constant and sustainable information to diabetic patients about the potential risk factors for diabetic nephropathy such as hypertension, poor glycemic control, as these factors were identified in this current study. Acknowledgement. I am grateful to the conference organizer for this opportunity to present my research. My heartfelt thanks to my mentor, Dr. Tahira Jabin, the medical staff, and the patients in Hyderabad and Jamshoro for their support and the cooperation. Thank you for your attention.
Thank you so much for your valuable information. Now, I would like to invite Savera Dilbar from GCUF to talk on the topic of single high dose streptozotocin treatment severely damages kidneys in adult Western rats. Please come and share your words with us.
Assalamu alaikum. My name is Vera. I am from Government College University, Hyderabad. My presentation topic is single high dose streptozotocin treatment severely damage kidney in adult Western rats. Streptozotocin and alloxan are essential and commonly recommended diabetogenic agents. Streptozotocin has the versatility to induce both type 1 and type 2 diabetes. Naloxone is primarily used for induction of type 1 as a free radical generator, leading to DNA damage and subsequent cell death, and can be administered as either a single high dose or multiple low doses. Notably, STG is a highly selective cytotoxic agent that specifically targets and damages pancreatic beta cells, which are responsible for insulin production. Kidney. The kidney filter excess glucose from plasma utilizing renal glucose transporters located in proximal tubules of nephron to reabsorb the glucose. Objective. This study was conducted to investigate the effect of single high dose of STC on kidney function. Renal function was assessed through microscopic examination and biochemical analysis following STC treatment. Serum level of creatinine, blood urea, nitrogen, insulin, and hemoglobin were measured to understand the progression of diabetic nephropathy. Additionally, histological changes in the pancreas and kidney tissues were examined. Material and method. A total of 6 gram albino blister rats were obtained from the Department of Physiology at the Animal Research Station. Water and job maintenance were provided to every rat on an as-needed basis. The Government College University Hyderabad Animal Research Ethical Committee guidelines were followed in carrying out each and every experimental protocol. A single intraperitoneal injection of STC 200 mg was given to adult rats. Animals were sacrificed on multiple time points after STC treatment, 12, 24, and 36 hours. Blood glucose levels were measured by pricking the tail veins using glucometer. Tissue from kidney and pancreas was assembled at the designated hours of sacrifice, and it was permanently fixed in 10% formalin. Blood was taken in tubes and the plasma was obtained by centrifuging the tubes at 4000 rpm, and serum sample was collected in an Pandoff container and frozen at -20 Celsius. Graphs. This graph represents the urea and burn level at different time intervals after 24 hours, 12 hours, and 36 hours. Urea is a waste product in the blood, and burn test is used to measure the urea and nitrogen in the blood. Creatinine level. This graph represents the creatinine level at different time intervals 12, 24, and 36 hours. X-axis represent the time intervals 12 hours, 24 hours, and 36 hours post STC administration. Y-axis represent the creatinine concentration in blood. At 12 hours, creatinine level are lower compared to later time points. By 24 hours, creatinine level rise significantly, indicated the kidney filtration is becoming impaired. At 36 hours, creatinine levels continue to increase, reaching their highest point, suggesting progressive and worsening kidney damage. The increased trend of creatinine level from 12 hours to 36 hours suggests a time-dependent deterioration of kidney function due to STD-induced toxicity. Glucose level. This graph shows blood glucose level over time measured at three intervals 12 hours, 24 hours, and 36 hours. Y-axis represents glucose concentration ranging from 0 to 700. X-axis represents the time point after treatment on intervention after STC administration 12 hours, 24 hours, and 36 hours. At 12 hours, blood glucose levels are quite low. At 24 hours, blood glucose levels spike significantly. By 36 hours, blood glucose levels remain elevated, indicated the hyperglycemia persists. This graph effectively demonstrates the progression of hyperglycemia in animal model following STC administration, providing a clear timeline of how STC-induced diabetes developed over 36 hours. HP level. HP level almost remain unchanged after 24, 12, and 36 hours. This is the histological pictures of kidney. Kidney damage. Kidney tubular necrosis, damage to renal tubular cells leading to cell death. Glomerular damage, injury to glomerular capillaries, mesangium, and podocyte. Interstitial inflammation, infiltration of immune cells. Apoptosis means programmed cell death in renal cells. We can see here histological pictures of pancreas damage, damage to pancreatic beta cells, inflammation of pancreatic isolates, programmed cell death in pancreatic beta cells, reduce insulin staining. Conclusion. Blood urea nitrogen and serum creatinine levels were not comparable between treated group and control group. Microscopic examination revealed signs of tubular or glomerular damage. The currently study revealed that single high dose STD induced dose impact the kidney. Thank
14.1. And the aims of the study is the purpose of this research was to determine whether or not SNP is linked to the higher risk of breast cancer along with some known genetic factor in women from southern Punjab, Pakistan. And objective of the study to find genotypic frequency and allelic frequency in both patients and control, and to study association of risk factor including marital status, parity, family history of any cancer, and area of residence, any other disease, and body mass index. To determine the estrogen receptor, progesterone receptor, human epidermal growth factor in breast cancer patient is also the objective of this study. This flow chart of the study design indicate the population of current study, that is 190, and the control in which control is 90 and breast cancer patients are 100. Data are collected by questionnaire by face-to-face interview, and these data are arranged in Excel and analyzed by SPSS software. and the sample are collected from the Minar Cancer Hospital, Multan. And this after the sample collection, the DNA isolation was done by manual method, that is salt and precipitation method, and then gel electrophoresis will be performed. And materials and methods in which DNA extraction procedure was done manually by salt and precipitation method. 3 ml blood was taken from both patients and controlled. And after DNA isolation, when all the DNAs are extracted, the PCR was performed to analyze the studied SNP, and the genotyping of this gene was done by PCR. This is efficient and reliable and low-cost method, and the result of the PCR are shown in the electrophoresis gel. And these are the tetra-arm PCR for genotyping. The primers were designed, and these are the sequence of the primers, and they are the product size. T allele is 253 base pair, C allele is 199 base pair, and outer band has 396 base pair. Next, comparison of breast cancer patient and control group based on demographic factor. These are some general risk factor: parity, marital status, history of any other cancer. Parity is also subdivided into subgroups, into 3 subgroups, and their frequency and percentage in both controlled and patient are given here in this table. And the parity p-value shows that it is significant, 0.01. Any other cancer is also divided into 2 subgroups, positive and negative, and their p-value shows there is no weak significant, 0.095. And next, demographic factor is area of residence, also subdivided into 2 groups, urban and rural, and their percentages of patient and control are given in this table. And their p-value is 0.074. that shows that it is weak significant and not significant value. And any disease other than cancer is also divided into 2 categories, yes and no, and their frequency percentage in both patients and control are given, and their p-value indicate it is a highly significant because p-value is less than 0.01. And last is demographic factor is body mass index, also subdivided into weak, normal, overweight, obese, and their percentages are given in this table. And this shows that their p-value shows that it is a significant, 0.01. Results of PCR that are shown by this electrophoresis gel. This is 2% of agarose gel, shows that shows the result or SNP of patients. In this gel, first lane 100 base pair DNA ladder is present, and a heterozygous genotype was present in the lane 2 and 7, and the CC genotype, that is homozygous, shows in the lane 5 and 6, and TT genotype shows in the lane 1, 3 and 4. The outer band, we will also discuss this, is there is 396 base pair band, and T allele is 253 base pair band, and C allele is 199 base pair band. Allele candidate frequency of this SNP in both patients and control are given in this table. T allele is 96.5 in patient, but in control is 68.5. And in C allele in patient is 23.5, and in control is 121.5. And the genotypic frequency of both patient and control are present in this table, and their p-value is 0.286. And distribution of this SNP genotype among cases and control are shown in the graphical form. And this is the histopathological information of breast cancer patient. First one is estrogen receptor. Estrogen receptor is also categorized into 3 group: positive, negative, and unknown. Positive is 52%, negative is 39%, and unknown is 9%. And progesterone receptor is also subdivided into 3 subgroups. Negative is 44%, positive is 47%, and unknown is 7%. And human epidermal growth factor is subdivided into 4 categories: negative, positive, unknown, equivocal. Positive is 23%, negative is 61%, unknown is 13%, and equivocal is 3%. And in conclusion, the present study shows that this SNP was not significantly associated with an increased risk in breast
cancer in the southern Punjab population. However, other risk factors such as parity, marital status, body mass index, and any other disease other than cancer were significantly associated with breast cancer patient. Thank you for your attention, and that's all from my side.
Thank you so much for sharing your knowledge. Now, I would like to welcome Kainat Arshid from GCUF to talk on the topic of repeated low dose alloxan treatment fails to induce tubular damage in adult visitor rats. Please come and share your knowledge on the topic with us.
Assalamualaikum. My name is Kainat and I am from Physiology Department, Varun College, University of Pakistan. My presentation topic for ICAZ 2024 is repeated low dose of naloxone treatment fails to induce tubular damage in adult blister rats. Naloxone kidney filter out excess plasma glucose. Kidney play a crucial role in excretion by filtering out excess uh, plasma and the waste products in the form of urine. Naloxone, a beta cell cytotoxic chemical, is commonly used to induce hyperglycemia in rodent model. Naloxone induces the formation of reactive oxygen species within beta cells, and reactive oxygen species are highly reactive molecules that can damage cellular structures, including DNA, protein, and lipids. Aloxan, it is commonly known as alox, and its complete name is aloxanum, and it is an organic compound that is synthesized in the laboratory. And the sphere of action of aloxan are pancreas, liver, kidney, blood, and lung. But the main organs that are mainly effective and affiliated by the lockdown, these are pancreas, kidney, and liver. The necrosis occurs specifically in beta cells due to their selective uptake and accumulation of alloxan compared to the cells. Uh, Alloxan is a mimic glucose. It mimics glucose and it is a glucose analog. Uh, when glucose is uptake by the glucose transport cell, the alloxan is also this is also taken up by the glucose transport cells on the pancreas, and when they enter, they are when they enter pancreas, they selectively damage beta cells. When they will damage beta cells, they will cause the production of reactive oxygen species that will ultimately cause production of oxidative stress. And oxidative stress, these itself cause halt many physiological functions, halt many physiological functions. And when the beta cells will destroy, they will be unable to produce insulin, and there will be no insulin. Condition occurring to the body called type 1 diabetes. The present study was designed to study the impact of multiple repeated low dose of naloxone on the kidney. Our aim for this study was to, because I have mentioned before that the naloxone have mainly effect along kidney, liver, and pancreas. Our aim was to investigate that how much the repeated low dose of naloxone have damage to the kidney, pancreas. So we evaluate renal function post-naloxone treatment through microscopic examination and biochemical assessments, and measure serum levels of creatinine, blood urea nitrogen, insulin, hemoglobin to understand the progression of diabetic nephropathy, and examine histological changes in the pancreatic and kidney tissue. We took material method. We took a lesson from the health biology lab of Government College University Faisalabad, and the rats, six rats, we made six rats from the research station of the Department of Physiology, 180 to 200 gram uh, rats that weighed 180 to 200 grams were obtained, and they were acclimated. They were acclimated for 1 week and at a temperature of 26 degrees Celsius and 40 to 60% humidity, and water and maintenance was provided to every rat, and intraperitoneal injection of low-dose 40 mg was used to induce diabetes for 4 days in a row, and uh, nicotinamide was also injected after post-aloxin treatment, and after 24 hours, the blood glucose level was measured. We measured blood glucose level by pricking the tail vein using glucometer, and rats having blood glucose level higher than 180 grams were considered diabetic. And the animals were sacrificed on multiple time points at post-aloxin treatment in 12, 24, and 36 hours. The blood samples were assessed for CBC, serum creatinine, blood urea nitrogen, and the kidney and pancreas were also collected and fixed for histological analysis. The blood urea nitrogen and serum creatinine levels were comparable between the treated group and the control group. Pause. As I have mentioned that the results were comparable because as you can see that there is a spike in the blood glucose level, but the plasma level of blood urea nitrogen and urea and the creatinine levels, these all are comparable. There is no big difference. So this is the histological microscopic picture of kidney. As you can see that there is a small amount of damage to kidneys and pancreas. The pancreas and kidneys are not effective and their function is intact. The repeated low dose of alloxan do not cause huge damage to the pancreas and kidneys. The blood urea nitrogen and serum creatinine levels were comparable, and the microscopic examination reveals no sign of tubular damage. Uh, the current study reveals that repeated low dose of alloxan does not impact kidneys. Thank you.
Thank you so much for your information. Now, I would like to invite the sixth presenter of the session, Iman Khalid from GCUF, to talk on the topic of renal toxicity is induced by a single high dose of alloxan in adult Vista rats. Please come and share your knowledge with us.
Assalamualaikum. I'm Iman Khalid. My presentation topic is renal toxicity is induced by a single high dose of alloxan in adult blister rats. Alloxan. Alloxan, a beta cell cytotoxic chemical, is commonly used to induce hyperglycemia in rodent models. Alloxan's diabetogenic activity is mainly due to its ability to generate reactive oxygen species. It induces oxidative stress within beta cells, leading to cell death. Alloxan enters the beta cells via the GLUT2 glucose transporter. Alloxan might be absorbed by the proximal tubules of the nephrons via GLUT. Alloxan is commonly used to induce type 1 diabetes in experimental animals such as rats and mice. Kidney. Kidneys filter out excess plasma glucose. Renal glucose transporters present in the proximal tubules of the nephrons are employed for the uptake of glucose. Material and methods. A total of 6 albino rats were obtained from the Department of Physiology at the Animal Research Station. Water and chore maintenance diet were provided to every rat on an as-needed basis. The Government College University in Faisalabad Animal Research Ethical Committee's guidelines were followed in carrying out each and every experimental protocol. A single intraperitoneal injection of alloxan was given to adult rats. Animals were sacrificed on multiple time points after alloxan treatment, that is 12, 24, and 36 hours. Blood glucose levels were measured by pricking the tail vein using glucometer. Tissues from kidney and pancreas was assembled at the design and it was promptly fixed in 10% formalin. Blood was taken EDTA tubes and the plasma was obtained by centrifuging the tubes at 4000 rpm and the serum samples was collected in an Eppendorf container and frozen at minus 20 degrees Celsius. Results. This graph shows the urea and burn levels at different time intervals after alloxan treatment induced by toxicity. These markers help to understand the severity of kidney damage induced by alloxan. This graph shows the creatinine levels at different time intervals after alloxan treatment. This correlates with the progression of nephrotoxicity as the kidney lose their ability to clear waste products efficiently. Results represents the glucose levels at different time intervals after alloxan treatment. It represents excessive hyperglycemia induced by alloxan. Hematoxylin and eosine stained kidney tissue from single high dose of alloxan treated rats. This image shows the tubular dilation, interstitial edema, and glomerular hypertrophy, which all of which are hallmarks of kidney damage induced by alloxan. Hematoxylin and eosine stained pancreas tissue from single high dose alloxan treated rats. This image shows the beta cell necrosis, which are caused by alloxan-induced toxicity, which leads to diabetes by reducing insulin secretion. Conclusion. Increased blood, urea, nitrogen, and serum creatinine levels were observed in the alloxan-treated rats. Microscopic examination reveals glomerular hypertrophy, increased extracellular matrix deposition, and tubular dilation. The current study reveals that high-dose alloxan induces acute glomerular hyperfiltration, tubular toxicity, and inflammation, which are independent of hyperglycemia. Thank you for your attention.
Thank you so much for your valuable information. Now, I would like to welcome Isha Razak from GCUF to talk on the topic of use of different cost-effective animal BT products to replace fish meal, effect on growth performance, nutrients digestibility, and hematological indices of major carbs. Please come and share your knowledge on the topic with us.
Assalamu alaikum. Myself Isha from Government College University Faisalabad from Department of Zoology MPhil. Today I am presenting my topic use of different cost-effective animal byproducts to replace fish meal, effects on growth performance, nutrient digestibility, and hematological indices of major cause. First of all, we discuss about need of this project. As we know that human population and their living standard are increasing day by day and cause scarcity of proteins. Fishes are the major source of animal proteins for humans. It directly lead to increase the pressure on aquaculture industry. Fish meal is the major source of protein for fishes, but it is most expensive and unsustainable source. There is a high need to find substitute of fish meal which have similar palatability. For this purpose, different plant protein sources are widely used in aquaculture. Because of their great availability, favorable nutrition profile, and competitive pricing, plant-based proteins offer a interesting alternative. Now we'll talk about animal byproducts, which we replace with fish meal. Animal byproducts are being preferred because they have higher amount of protein and lipids, essential amino acids, and better digestibility. There are some animal byproducts being used in aquaculture nowadays. Feeder meal, blood meal, insect meal, poultry byproducts meal, meat and bone meal. All of these are used as alternatives of fish meal. Because of fish meal is expensive and unsustainable. Next, we talk about why animals byproducts are better than others. Blood meal is a composed of most 80% of protein content, so it is a better alternative. Poultry byproducts meal is obtained from ground or clean parts of the carcass of slaughtered poultry. Meat and bone meal is an animal byproduct which is obtained from different slaughtered horses. Good nutritional value of insect meal is due to amount of proteins which varies from 52 to 80% in different insect species. There is a low carbohydrates content, satisfactory amino acid profiles, and low phosphorus content in animal protein source, so they are referred to as better substitute as compared to others. Now there are fishes which are mostly used for eating. Indian major carps are the most important fish with maximum market value demand by consume due to their better taste and fish flesh. They contribute about 60% of total freshwater fish production. The major carps are most preferred from fishes because of their fast growth and high acceptability to consumers. Indian major carps are Catla, Libiorhita, and Sphinx murida. Here is ingredient diet composition table. We can see that animal byproducts are mostly used and have better results, as you see in this table. Next, materials and method. Fish and experimental conditions, feed ingredients, and diet formulation, formulation of feed pillars, feed protocol and sampling, proximate analysis, growth assessment and nutrient digestibility, hematological parameters, and last statistical analysis. Then we talk about growth study. We use some formulas such as weight gain, FCR, and SCR. We can use these formulas to find our growth. As you see in these figures, all of these figures show that insects meat show better result as compared to other animal byproducts or feeds. There is diet control plan, diet 1, 2, 3, 4, 5, 6. In this figure, we easily show that, see that insects meal is better than all of others. Here, hematological parameters as red blood cell, HB white blood cell. In With this vision, we say addition of animal byproducts could enhance the growth, digestibility, tarsus composition, and blood profile of Civis mirigala. Other byproducts are good, but no show good results as we think. Due to many reasons, because of environmental conditions, fish species palatability, they have greater advantages over plant-based protein sources, such as complete amino acid profiles, high level of lysine, methionine, and phosphorus. Some of them. That's it. Any question? Thanks for your attention.
Thank you so much for your valuable information. Now we will proceed toward the eighth and the last presenter of the session, Zinat Nasir from GCUF, to talk on the topic of minimal renal impact of repeated low dose streptozotocin in adult visitor rats. Please come and share your knowledge on the topic with us.
Assalamualaikum. My name is Zinat Nasir. I'm here to represent on the International Conference of Applied Zoology 2024. The topic I am from Department of Physiology from Government College University of Faisalabad. Today I am here to represent my thesis research topic minimal renal impact of repeated low dose streptozotocin in adult visitor rats. First of all, I am I'm here to explain about the streptozotocins. Streptozotocin and alloxans are the diabetogenetic drugs. These drugs are given to the rats to cause diabetes. Streptozotocin can be used to induce either type 1 diabetes or type 1 or type 2 diabetes, where alloxan is commonly used to induce type 1 diabetes. It is highly selective cytotoxic agents that specifically target fundamental diabetes. Low dose injections. Kidney filter out excess plasma glucose. Renal glucose transporter present in the proximal tubules of the nephrons and employed for the uptake of glucose. So we can discuss here about how streptozotocin enter into the kidney. Low dose of streptozotocin can enter the kidney throughout the bloodstreams after being administered, usually are injections. STZ is typically given through intravenous or intraperitoneal injections. It enters the systematic circulation and is distributed throughout the body. Once in the bloodstreams, STZ travels through the circulatory systems and reaches various organs, including the kidney. The kidney filter bloods to remove waste products and regulate fluid balance. STZ, being a small molecules, can be filtered through the glomeruli. Glomeruli is the filtering unit of the kidney. After filtration, some of the STZ can be absorbed in the renal tubules depending on the concentration and condition within the kidney. In the kidney, streptozotocin can selectively enter renal cell, particularly those in the proximal tubules. The exact mechanisms involve specific transporters which can facilitate the uptake of the drugs into the cells. Once inside the renal cells, mainly the high dose of streptozotocin can exert into a toxic effects, contributing the oxidative stress and inflammations, which may lead to the damage and dysfunctions over the times. The main objective of this research is to present study was designed to study the impact of multiple repeated low dose of streptozotocin on kidney and to evaluate the renal functions using microscopic inspections and biochemical analysis, and also assesses the serum levels of creatinine, BUN, for the development of diabetic nephropathy, to look at histological alterations in the tissues of the pancreas and the kidneys. Then there is a material and method. 6 Vista rats, adult Vista rats, and their weight are around about 180 to 200 grams, which we were obtained from the Animal Research Station Department of Physiology. Their rats were given CMP diets and waters. All the experimental protocols were performed taking into the direction of Animal Research Ethical Committee, Government College, University, Faisalabad. Diabetes was induced by administering intraperitoneal injections of low-dose streptozotocins was given 35 mg over kg for 4 days consecutively. These rats were given intraperitoneal injections of fresh diluted nicotinamide 15 minutes after receiving intraperitoneal streptozotocin treatments. Then there is a 24 hours after last injection, blood glucose levels were measured by pricking the tail vein using glucometers. Rats having blood higher glucose level than 180 were considered diabetic. Glucose was mainly monitored by glucometers. All the rats were humanly euthanized, and blood and tissue samples were collected for further biochemical and histological analysis. Then pancreas and kidney tissue samples were collected in 10% formalin solutions. These tissues were collected for the histological examinations. In histological examinations, further many stages are involved, in which first we collect the samples of the desired tissues, then there is a fixation, washing, dehydrations, then there is cleaning. After cleaning, fax infiltrations, embedding, sectioning, mounting, and staining. These methods are for the histological examinations. Then we get a result. Here is the plasma creatinine levels of control and treated values, and hemoglobin. It is the mean plasma level of blood urea nitrogen, which is also called burn and urea. Mainly in this topic, in this research, streptozotocins can cause diabetes, but it cannot affect on the kidney. The test results were not changes. Here is the slides of blood glucose level, in which blood glucose levels are rises above to 180, and these rats are also diabetic. Histological picture of control and treatment groups. Then there is no effect on these pictures. In conclusion, blood urea nitrogen and serum creatinine levels were comparable between the treated groups and the control groups. Microscopic examination revealed no signs of tubular and glomerular damage. Study reveals that the low dose streptozotocin did not impact on the kidney. Thank you for listening.
Thank you so much for sharing your knowledge on the topic. Now, I would like to thank all the presenters and all the participants to attend 7th International Conference on Applied Zoology 2024. This was our last presentation.